Thanks, Vicky, for the introduction. So, as Vicky said before, my PhD is in neurobiology. And one thing I always found fascinating about neurobiology, or let's say clinical neurology, is of all the disease models and organ systems of the human body, in the last 100 years, whether it be the heart, the kidney, the liver, any infectious disease, we've made immense strides in understanding them as well as treating them. However, diseases of the nervous system, of the human mind, is the one we've made the least advancement in. And I find this rather odd, considering the nervous system, and specifically the human mind, is what makes us different from any other animal species. Our ability to learn, our ability to recall memory, have emotion, have language, communication, love, affection, social interactions. It's really remarkable. Yet, diseases of this organ system are the ones that we've made the least advancements in. And they affect all human beings at all ages, whether it be dementia in the elderly. Think about your grandmother or your mother not remembering who you are. Or people in their middle of their lives. Think about PTSD. Think about bipolar disorder, manic depression. And children are no exemption. Neurodevelopmental disorders include things like cognitive impairment in the form of mental retardation, or in the form of autism, such as having impaired language or impaired social communication and understanding of social interactions and emotions. And autism is one of these diseases that's quite fascinating, in my opinion. It's simply the most commonly occurring cognitive or neurodevelopmental disorder in children and in all humans around the world. In the case of the Western industrialized world, in the OECD, autism occurs in 1 in 2,000 live births. And an interesting fact I realized was a couple of years ago, a lot of people are bringing up the point that autism is a disorder of the West, of the industrialized world. We have too much money, we have too much processed food, so we came up with a new disorder, something that affects our children in the West. Oddly enough, an interesting statistic, autism has a 3 times more higher prevalency in low and middle income countries. Why could that be? One of the common reasons is consanguineal marriages, poor environmental health and toxicology, poor nutrition. It's a global disorder. And diseases of the nervous system are actually the single most stigmatized disorders in societies all over the world. Think about people being ostracized for not being able to learn, for not being able to speak properly, for not being able to socially interact. And it's really quite strange that this disorder, or any type of disorder of the mind, especially disorders of children in their behavior, such as autism, are so poorly understood. They're poorly diagnosed and poorly treated, and we tend to just treat the symptoms. And as a neurobiologist, this is one of those things that I always wanted to understand. Why and what can we do? And the most common form of pediatric cognitive impairment is autism. Now, autism has a problem in that it's really a spectrum disorder. It's not diagnosed, unlike all the other disorders mentioned earlier, by a single lab test for a marker. It's really a behavioral diagnosis. According to the DSM-5 of the American Psychiatric Association, there are 2 major metrics that must be met. Lack of or poor social communication, which can be understanding of emotion, language, and the second aspect is engagement of repetitive behaviors. And autism spectrum disorder has a mass spectrum in terms of the severity and also subsets. But there's one subset of autism spectrum disorder, which is called Fragile X syndrome. And this is why I study. And Fragile X syndrome is simply the most commonly inherited form of cognitive impairment, and it encompasses all the criteria of autism, including intellectual disability, delayed onset of language acquisition and communication, anxiety, aggression, epilepsy, and engagement of repetitive behavior. But one key feature of Fragile X syndrome compared to other forms of autism is that it has a known genetic basis, or a monogenetic basis, meaning we know that there's one gene that's simply implicated in the manifestation of this disease, unlike other forms of cognitive impairment, and autism spectrum disorder in particular. And what we know about it is this gene is located on the X chromosome, and it has in its regulatory region upstream, called the 5' UTR, a CGG repeat or a triplicate repeat. This repeat is kind of like the on-off switch, think about it, for the gene. And in normal human beings, we have anywhere from 5 to 55 of these upstream of that gene. In the case of Fragile X syndrome, we have over 200 of these repeats. And what happens is when we have over 200 of these repeats, there's a biochemical change that occurs at the nucleotide, specifically at the cytosine residue. And this gets methylated, and this causes a change in the nucleotides and the chromatin that organizes that gene. And this results in the lack of the expression of that gene, resulting in no production of its RNA and its functional protein called FMRP. And the analogy I like to think about when understanding this disease is the FMR1 gene is kind of like a light bulb that goes on and off. When the light bulb goes on because it has power from a power line when it's connected to a house. When it's on, the CGG repeat upstream of it is basically allowing it to turn on. So we can turn the switch on and the light goes on. And when it's methylated, it's the off switch basically, and this gene is turned off. Now, in the case of Fragile X syndrome, it's been heavily modeled in the mouse historically, and there was one main reason for that. We can't ethically do any type of testing on a human being. So the mouse was the next best model, and it's been the favored model of neurobiologists to model diseases. But there's one little problem. The mouse has the same gene structure as the human FMR1 gene. However, when scientists have tried to genetically modify the CGG repeat upstream by expanding the amounts, the gene does not actually turn off. The gene transcript is produced and we still get the functional protein, which is problematic. And then what happened was scientists instead decided to delete one part of the gene, the exon 5, and then use that to study the mouse model. Now in this case, when this part of the gene was deleted, we didn't receive any functional protein, and mice actually have autistic-like behavior. And this was great for studying the disease for about 30 years. But in my mind, this is kind of like the light bulb analogy where you say, you have the on-off switch, but it doesn't matter if the switch is on or off, the light always turns on. And we try to understand principles from this on-off switch for the human and transfer it there. Well, what happens if your light bulb also has an extra power supply because it's connected to a solar panel? And that's how I like to think of this analogy. So people have used this model of the mouse to model Fragile X syndrome for 30 years, and about 90 clinical trials have been based off of mouse data. Not one single one passed phase 1 clinical trials. And basically that's because the mouse model has a completely different mechanism of how it manifests, and that's really the epigenetic change. So that means that this disorder really has a very key epigenetic factor, which means, beyond the gene itself, that we need to understand. And my co-supervisor, who will speak next, Professor Nissim Benvenisty, really was very revolutionary in understanding this. And in 2007, he made the first human embryonic and later the first induced human embryonic stem cell line from Fragile X patients. And what he did was he took wild-type fibroblasts. So fibroblasts are kind of like this connective tissue beneath the skin. And he took these wild-type fibroblasts and then as well as fibroblasts from Fragile X patients, and reprogram them to turn them into human stem cell lines. So here in this figure, we see that the wild-type fibroblasts have a high expression of this FMR1 gene. The Fragile X ones have no expression, and many reprogrammed ones that were turned into stem cells don't either. And that was actually the first model that we could actually use to model Fragile X syndrome in humans accurately, and to do things like drug screens and identify compounds that could possibly reactivate this gene. And in a subsequent study from his lab, he performed a screen where he identified one compound called 5-azocytidine. And 5-azocytidine, when given to Fragile X stem cells, massively results in the increase in the FMR1 gene transcript at the RNA level. And also, neurons that are derived from these same Fragile X iPSCs also have increased FMR1 expression. And this was fantastic. It was finally the first time someone found a compound that actually works on the human system. However, there's one little caveat if we want to bring this to clinical trials or to clinical level. 5-AZOC is highly toxic and stays in the system, and in particular in highly dividing cells. So imagine having a child who has Fragile X syndrome and you're giving them something that functions to some extent like a chemotherapy agent. So for proof of principle, it's great, but in reality for use, it's another story. But we were thinking, what are some possible ways to target this methylation? And this methylation is actually the key aspect. So 5-azacitidine actually targets what are called DNMTs, or DNA methyltransferases. And what they do is they add a methyl group to the cytosine residue, or that cytosine nucleotide. And the 5-AZOC blocks that function and prevents that addition. One thing we were thinking about for this project with my co-supervisor Verdon Taylor and Nissim Benvenisty is, instead of preventing the addition of DNMT, why not actually promote the removal of the methyl group by an enzyme called the TET enzyme? And this is actually kind of also more important because I'll get to it next, is that DNMTs primarily work in cells that are dividing. And neurons, which are the major component of our nervous system, actually don't divide. And therefore, DNMT activity is not really viable. However, TET enzymes are highly active in the nervous system. So we thought this could be a potential druggable target. And TET enzymes really catalyze the removal of methyl groups in a series of many steps. And as you progress along these reactions, it allows the gene that has this methylation to be gradually more active transcriptionally or produce more of that protein. And the great thing about TETs is they can be pharmacologically manipulated, they work independent of DNA replication or cell division, and they have high function of neurons. So our hypothesis was, if we have methylated CPGs or groups of nucleotides in that 5' region, this doesn't allow any transcription of the FMR1 gene, and there we have Fragile X syndrome. However, if we can give some of the substrates that either activate the TET enzyme naturally or by modulation, we could promote the recruitment of the TET enzymes to this island and basically cause erasure or removal of this methyl group. And two major components that activate the TET enzyme are vitamin C, which we get in our orange juice, and also a metabolic intermediate called alpha-ketoglutarate. And our idea was, if we can just flood the system in culture or in an animal with this, could we promote the erasure of some of these methyl groups and promote the transcription and production of the functional product, FMRP. So it's really a simple experiment what I did. I took Fragile X stem cells, cultured them at a very low density, and then administered vitamin C for about 6 days in culture, and then would collect the stem cells and then do quantification and measurements of the gene expression. And actually what we saw was a dose-dependent increase in the expression of FMR1 in Fragile X stem cells. And actually at the higher levels than with AZOC, which was done previously. So this was quite promising. So it was great. Vitamin C can promote FMR1 re-expression in Fragile X iPSCs. But we needed to validate this in a neuronal system of humans. So we decided to do what was called an organoid protocol, or make a pseudo brain in culture. We would take the Fragile X stem cells and we would culture them in a way where they don't stay on a flat surface, but rather are constantly moving and they form a ball, an aggregate. And you could call it like mimicking to some extent development of the human nervous system in the uterus, in utero. And we would follow this protocol, which was discovered about 11 years ago by the lab of Jürgen Knoblich. And then we decided to do this and administer vitamin C and just look at things like gross morphology. So we categorize the organoids into 3 categories. One, which would be called the gold standard organoid. They have nice, smooth morphology, they develop nicely, and phenotypically, they just look good. Then there's another category which we call category 2. These are organoids that they have some good nervous system, but they also have a lot of tissue that didn't form the nervous system. They became like little vacuoles or sacs of skin. And then we have the third category, which are the entirely cystic organoids. So upon treatment of vitamin C for these organoids, we noticed that compared to a wild-type control, most wild-type organoids were in category 1 and 2, about 80% of them. And very few didn't develop correctly, maybe about 10 to 15%. On the other hand, Fragile X organoids, as you can see here, not as smooth, not as pretty, highly cystic. It's actually about 10% were decent or very good, and 90% were terrible, didn't develop at all correctly. And upon vitamin C treatment, the majority of them actually look like this, quite well developed, maybe a little immature for their proper developmental stage, but when we quantify, massively increased and shifted. So that indicated that we got kind of some degree of rescue. And then what we were very interested in seeing was this methylation switch, this on-off switch. Does that really change or not? So in the gene, there's these CPG islands, which are upstream of that 5' UTR, which are kind of a region that's not so repetitive that we can't identify it, but it's kind of close enough and unique that we can actually identify and characterize it. So we were very interested to see in this area what percentage of this island had a methylation. So if it was completely Fragile X, it would be highly methylated. If it was wild type, it would have a reduced methylation. And then we saw that compared to, when we look at Fragile X organoids, they're mostly about 80 to 85% highly methylated in that region, versus the wild type, which are the normal ones, which are about 10%. And a substantial amount of them have, 3 of them actually out of 7, had a substantial rescue, about 40%. So we got some erasure, which was great. And then there was also a percentage that didn't respond. And this was done in collaboration with Faiza Noreen, a postdoc specializing in methylation in the lab above us, in Primo Scher's lab. So this was great. We actually saw that vitamin C promoted demethylation in the CPGs upstream. So basically, thus far, what we did was we established the Fragile X organoids and showed and demonstrated that vitamin C treatment does cause significant increase in the FMR1 expression level in the Fragile X iPSCs, and that treatment could actually rescue the neuronal tissue in the cerebral organoids. And lastly, that it actually caused a demethylation in those islands as well. So finally, I'd like to thank the BRCCH for funding this program and for giving us the opportunity to do this work. And of course, I had 2 excellent labs to work with, Nissim's lab in Jerusalem, where I went for my first year to actually start this project and establish it, and then in Verdon's lab, where I've been based for the last 2.5, 3 years as well, working on it. Faiza was incredible with helping with the pyrosequencing and the methylation, as well as many of the members of the Scher lab. Kangsha was helping me with the organoid protocol from Barbara Treutlin's lab at the DBSSE here in Basel, and the Timper lab for helping with some of the metabolic aspects of the project. So, thank you.